everyone, we are from Group 5 and would like to share about the enhanced fish diversity following active coral reef restoration efforts in Tun Sakaran Marine Park at Sabah, which is also the Malaysian Coral Triangle. This is our group member list, composed of 5 members as shown at the right side.
Coral reef ecosystems are vital to both ecological and human communities, providing essential services such as coastal protection, fisheries, and tourism revenue. Located in Malaysia's Coral Triangle, the Tun Sakaran Marine Park is a biodiversity hotspot that has faced significant threats from anthropogenic activities, including overfishing and climate change. In response to the degradation of these critical habitats, active coral reef restoration initiatives have been implemented since 2011, utilizing innovative methods such as coral frames.
So next, let's dive into the methodology part. From the Tun Sakaran Marine Park, TSMP, 2 coral reef restoration sites were selected to be surveyed. They are the depth end channel for Site 1, in which 30 coral frames have been placed since 2014, and Mandabuan Reef as the Site 2, where a total of 125 coral frames have been placed since 2011. The coral frames were placed at shallow zones from 4 to 10 meters deep at Site 2 and only at a single depth of 10 meters at Site 1. The field survey was done at this depth range too. It has been carried out 12 times starting from 27 July 2020 until 6 January 2021 during daylight hours between 8 AM to 3 PM. There are 3 major instruments for this study. First, the baited remote underwater videos (BRUV) was deployed for 60 minutes near the artificial reef within each study site. Second is the action camera, which was attached 1 meter in front of the bed, was used to record all the videos of fish found in this study. Lastly, the PVC quadrat was applied to guide the photos from the top of the coral frames placed next to the deployed BRUV. There was total of 25 photos taken from each of the study sites. For the section of data analysis, the fish seen in the video were identified at first to the lowest taxonomy level possible by referring the fish-based websites and published books. Then, relative abundance of the identified fish was also calculated using MaxN, which is a metric of species local abundance based on the maximum number of individuals observed in a single frame of video. These data were then calculated to get the diversity index using the Shannon-Weiner Diversity Index. Cruscal-Wallis test was also performed to compare the reef fish diversity between both study sites. Besides, the substrate cover on the coral frames were also determined using coral point count with Excel extension CPCE software. For instance, the substrate types were categorized into standard reef check guidelines such as hard coral, soft coral, nutrient indicator algae, rubber, sand, and others. Finally, the data were then further analyzed by calculating Pearson correlation index to determine the relationship between the percentage of hard coral cover on coral frames versus diversity of reef fish, and secondly, the percentage of hard coral cover on coral frame versus relative abundance of reef fish.
Based on the result obtained for fish abundance and diversity, total of 3,208 fish spanning 146 species from 27 families were recorded. And the most abundant families were Cassionidae, Pomacentridae, and Labridae. Meanwhile, moon wrasse was the most abundant species, which is 11.47% of observation. And from the table, we can see that the fish abundance and species diversity in Site 2 is greater than Site 1.
Examining the graph of substrate type, we observed that hard coral cover was notably higher at Site 2 at 37.96% compared to 27.7% at Site 1. This indicates a better establishment of coral fragment at Site 2, contributing to a more complex habitat for reef organisms. Soft coral cover was more prominent at Site 1. This is due to that coral fragment creating a substrate conducive to soft coral colonization. In contrast, Site 2 show higher proportion of rubbers and sand, suggesting limited coral growth and less effective maintenance of coral frame. This presence of nutrient indicator algae and sponges, touch minima, highlight potential challenge as their substrate can compete with coral. Overall, the difference in substrate composition between the 2 sites suggests localized environmental factor and maintenance efforts significantly influence restoration outcomes. Analyzing the correlation graph, we can see distinct pattern between coral cover, fish diversity, and abundance. A positive correlation exists between coral cover and fish diversity, as illustrated in graph C and D, due to increased habitat complexity from denser coral frame. These support diverse reef species, providing them with shelter and food resources. In contrast, graph A and B reveal a negative correlation between coral cover and fish abundance. This result could stem from insufficient coral cover to support higher fish population or the influence of other habitat types like rubber or sand, attracting fish away from coral frame. Site-specific difference also play a role. While Site 2 had higher coral cover and diversity, Site 1 exhibit greater fish abundance, suggesting the need to consider localized factor in future restoration effort. This finding underscore the importance of increasing coral cover to achieve both high diversity and abundance.
As for the discussion, it stated that the coral frame in this project can plant about 60 to 90 coral fragments in a time, and the preferable coral type is the branching, including Acropora subspecies and Poecilopora subspecies. The maintenance of the coral frame will be done on a regular basis, early of the month of the deployment, and the frame will be cleaned from the algae and that fragment will be replaced. Once the coral can grow healthily, the maintenance will stop. The correlation between hard coral and the fish diversity shows a positive correlation. There are 4 species of rays that are recorded at the frame, and 2 of it, which are spotted eagle rays and broad cow-tailed ray, is categorized under vulnerable category in the IUCN Red List. Besides, there are 2 additional species that are spotted in the video using BRUVs, spotted roaming around the artificial frames. Those rays are recorded associated with the ongoing coral restoration and rehabilitation projects that are happening in the site of studies. On the other hand, the hard coral cover and the abundance of fish shows a negative correlation. The planted reef does not show a major significance to the fish abundance in the area. However, other studies that are using natural reef shows a different result, which is positive correlation. As for now, the finding is maintained since the difference might be due to the lack of field data collected due to the MCO and the different structure and complexity of the reef. The challenges faced by the Sabah Parks Management in the restoration project, including manpower, logistics, and budgets.
In conclusion, the study conducted in the Tun Sakaran Marine Park highlights the importance of active coral reef restoration in enhancing fish biodiversity and abundance. With big challenges such as low hard coral cover on restoration structures, the presence of diverse fish species indicates a positive trend towards ecosystem recovery. The findings underscore the need for continued monitoring and adaptive management strategies to refine restoration techniques. By leveraging lessons learned from this research, park managers can better implement coral rehabilitation programs that not only restore marine habitats but also support local fisheries and communities. This initiative is crucial for ensuring the long-term health of coral reefs in Malaysia's Coral Triangle and safeguarding their invaluable contribution to both nature and society.
Here is our key reference article, and that's all from us. Thank you!